All right. So, as I mentioned before, this is going to be the first of two videos. This PowerPoint and video is going to concern B cell development. And you're going to see a lot of similarities here to B cell development as you saw with T cell development, except, there's going to be a few different things. Very most importantly, that B cells develop inside the bone marrow and they're continuously made throughout someone's life because they are developed in the bone marrow, and you never lose your marrow as you do with the thymus. So that's going to be one big thing. And the other thing is that the cells in the stromal aspects of the marrow do not also contain the same antigen presentation function as you would find in the thymus. So the B cell related autoimmunity checks of B cell receptor and their ability to see or sense human antigens is not nearly as broad or as in depth as you found in T cells. So typically how B cells develop, it comes in basically two distinct sets of phases, but in six different steps. And the first three happen in the marrow, and this is more to do with development. And so the first phase is very similar to what we saw with T cells. This is the generation of clonally diverse expressed B cell receptors as a function of VDJ recombination, and they are checked for their ability to bind to antigens and to produce a functional and stable supramolecular receptor. The second stage is negative selection. So this is inactivation or elimination of B cells that effectively bind to components of the human body. Again, this is part of central tolerance of the B cell. Again, it's not going to be as extensive as we see with the T cells, but it is a really important function of development. And then finally, stage three, and that is promotion of the fraction of B cells that are immature but produce a functioning non-human antigen recognizing B cell receptor that then gets released from the marrow and is then put into the secondary lymphoid tissues. That includes things like your appendix, your tonsils, and your lymph associated tissues like galt, malt, etc. Um, and then the second kind of component of development, which is more development via activation, happens in mature but naive B cells. And this again occurs in the secondary lymphoid tissue where B cells will mature um, and kind of circulate between the blood and the lymph. When there's an infection, they're drawn to the lymph, and when they are selected for, which we'll talk about later in the week, they then clonally expand and they then not only activate, but they also differentiate into plasma cells and memory B cells, which remain as B cells in the secondary lymphoid tissue. And the antibodies that are produced by that activation are produced inside the lymph nodes, but then released into the bloodstream and into the lymph. So again, we have initial stages of B cell development. This is going to occur in the marrow, and from the marrow, B cells will either basically survive in the lymph node, spleen, the tonsils, the adenoids, and the associate lymphoid tissue like the galt, Peyer's patches, the malt for the mucosal associated lymphoid tissue, so on and so forth. And they basically circulate in these systems because these are repository systems between the lymph and the bloodstream. So as B cells develop, when you have pluripotent hematopoietic stem cells, this is true for B cells and T cells, when you have the pluripotent hematopoietic stem cell, they express CD34. And again, this is a marker that can be distinguished on flow cytometry or looked for. And from CD34 positive pluripotent hematopoietic stem cell, again, these are all the stem cells that exist in the marrow. They start to express another biomarker, CD10. CD10 is a biomarker common to the common lymphoid progenitor. This is going to at least not completely move the B cell into a B cell derived origin, but it will determine fate cascade that once they express CD10, they can either become T cells, B cells, or natural killer cells. They have to exist as part of the lymphoid lineage. Um, as the cells produce CD10 and CD34, those that are that express LFA1 and other types of markers found on T cells that we previously discussed, they will find their way out of the marrow into the thymus. They'll home to the thymus by way of chemotaxis and cytokine expression. And B cells, because they don't get the same cytokine, they don't produce the same cytokine and chemokine receptors, so they have no draw or home outside of the bone marrow. And as they progress during development, they start to express CD127. And CD127 is a really important B cell specific biomarker, which is the alpha chain of IL-7 receptor. So IL-7, as we'll find out later, is going to be a really important cytokine for B cell development and B cell activation and function. And it's at this point when we're expressing CD34, CD10, and CD127, we are now producing a B cell precursor. This cell is now fated to become a B cell. And then finally finishing it off, they start to express CD19. We'll talk about CD19 throughout this lecture. And when you have CD19, CD34, CD10, and CD127, this is now a pro B cell, and we're now well on our way to producing a functioning, naive, but mature B cell. So the different stages of B cell development are as follows. You go from stem cell to early pro B cell to late pro B cell. And at this stage, again, this is where we have CD19, CD127, CD34, and CD10 expression in both of these segments. And in this segment, we get the heavy chain D and J are rearranged, and the light chain has not yet been established or changed. The late pro is where the variable segment is then combined to the diversity in the joining segment. So you get D and J rearrangement first, and then V is looped in. And again, the function of these is all based on differential expression of RAG, right? RAG 1 and 2. And so the expression of RAG and a few other important markers, including CD127 and CD34, are going to play, and CD19 are going to play a huge role in how this kind of arranges functional, spatial, and temporal expression of RAG and B cell specific genes. Once VDJ recombination has occurred and you create a functional heavy chain for the B cell receptor, this then is called a large pre-B cell, and you start to produce the mu chain of the immunoglobulin gene. So the VDJ component of the variable segment is then combined to the M component, right? So we have the heavy chain like so, and the light chain, and so you have so you have VDJ that has occurred in the variable segment of the heavy chain. The light chain has not been made quite yet, but you've now connected this to the mu constant region. So in reality, at this point, you have something that looks like this. So it's all just heavy chain with the mu FC region, and the fab region is just VDJ rearranged. So now, as you progress, once this is made and produces a functioning and stable, more importantly, stable, not functioning, but stable B cell receptor with a heavy chain, the VJ rearrangement is occurring in the light chain. This then gives rise to the functional B cell receptor, and then then we have the VJ in the heavy chain, the VJ in the light chain, the mu heavy chain, and the lambda or the kappa light chain, as it relates to, again, the constant region of the variable segment of the fab region. And so this is occurring here. And when we have gone through lambda or kappa, that's developed our constant region of our light chain, we then have a functional B cell receptor that can then be used for antigen recognition. All right, so as a B cell develops, it's going to express a wide variety of different receptors, and especially as it progresses from lymphoid progenitor cell to immature B cell. And this is going to become important because as the cell kind of develops in the bone marrow, it needs to have variable checkpoints in order to consider surviving. So as it survives, it's going to start expressing two really important factors, including things like ICAMs and integrins, as well as a important cell adhesion factor called KIT. And so KIT is a protein that binds to stem cell factor, and when KIT binds to stem cell factor, it actually triggers a phosphokinase relay that activates the expression of IL-7 and IL-7 receptor. So this triggers expression of IL-7 receptor, and for SCF, this triggers activation from the bone marrow to produce IL-7. This then allows expression of or is kind of the triggering event that then downstream activates a JAK-STAT cascade, which then activates the genes for pre-B cell development of the B cell receptor heavy chain. And then as the heavy chain continues to mature and show stability, it then activates the alpha chain or the light chain in order to develop a fully functioning immature B cell that is naive. So this is kind of the more or less the function of how this works. So the early pro B cell, again, it's going to undergo heavy chain rearrangement. The heavy chain rearrangement first rearranges the D and J segments. If the D and J segments are productive, this then allows for the variable region to be recombined. And so this happens on the first chromosome. If it works, then the B cell signals to survive. If it fails, this can also happen on the second chromosome. And again, if it works, then it's signals to survive. And again, this is really an important function because as this works, as we saw previously, when we get expression of IL-7, IL-7 receptor and continuous expression of IL-7 receptor is specific to the function of these eventually becoming a fully functioning heavy chain. If they don't, IL-7 expression is lost. And the end result of losing IL-7 is apoptosis of those cells. Now, it's important to note that as these cells continue to move along, the pro-B cell, especially to go between early pro-B to late pro-B and late pro-B onwards, is really important to continuously activate RAG. And RAG is actually expressed or regulated by two important transcription factors, E2A and EBF. So these two actually activate us. They are transcription factors that are activated through, again, the movement of going from early pro to late pro. And again, all of these are really part and parcel through the expression of IL-7. So the expression of IL-7 induces the expression of E2A and EBF. They activate a really critical transcription factor for B cells specifically called PAX5. And PAX5, as we'll see later, that it basically is a global transcriptional regulator for B cells that, among other things, is really important for activating the pro-B cell genes, immunoglobulin alpha and CD19, which is going to end up becoming the B cell co-activating gene, just like we saw with T cells in their expression of CD28. So in order for a pro-B cell to survive, again, everything happens in twos with lymphocytes. So they must produce a heavy chain that functions and a light chain that has the ability to bind the heavy chain. Now, this is the first two things that have to happen in order for the B cell just to survive. That has to happen before this is going to happen before it's going to interrogate human-specific antigens to see if it needs to be eliminated through that process. So the first thing is we end up getting the rearrangement of the heavy chain, and the heavy chain is then bound to something called V-pre-B. V-pre-B is expressed in relationship to the VDJ recombination in the heavy chain. So once the heavy chain has undergone VDJ recombination, that then triggers the activation of V-pre-B. So you get heavy chain VDJ recombination. It's productive. It produces a heavy chain that can be expressed. This then again activates and continuously expresses IL-7. This the beta chain, its co-receptors, immunoglobulin alpha and beta, and the expression of IL-7 all activate E2A and EBF, which the very first thing that it expresses in relationship to the expression of just the heavy chain in relationship to immunoglobulin alpha and beta is the expression of this V-pre B in lambda 5. And V-pre-B and lambda 5 basically are alpha chain segments that are non-recombined. So they exist in the alpha chain locus, but they exist as a functional light chain that is non-recombined. So it can serve as a surrogate, but does not have any rearrangement. So there's no recognizability. And all of the cascades that happen due to productive recombination and the expression of RAG haven't undergone anything. So this is just expression of a surrogate chain to bind to the heavy chain to stabilize it, to see if with an alpha chain does it actually with a light chain does it actually serve as a functioning and, more importantly, a stable supramolecular construct in order to pass the pro-B cell stage. So the V-pre-B and gamma 5 plus immunoglobulin alpha and beta and a fully recombined VDJ recombinant heavy chain all result in what we call the pre-B cell receptor. And it links into the membrane through a cytosolic component, which contains ITIMs and ITAMs. And there are disulfides that occur inside of the mu component. Again, we're starting with mu, and the mu has a single disulfide bridge in the hinge region in the C-terminal domain. So this, if it's productive, and most importantly, if this produces a stable molecule, and again, this is before we're checking for human antigens, this is just looking for stability, being able to signal into the molecule. The items that are associated with the cytoplasmic domain in relationship to a stabilized B cell receptor, pre-B cell receptor, actually inhibits VDJ recombination from continuing in the heavy locus chain and allows it to remove the remaining spliced out segments of the VDJ locus in order to maintain small and stable DNA at that locus, which can then be used to make conserved RNA throughout the different activation and generation of that particular B cell. So once the B cell receptor has formed in the pro B cell state, the pre B cell receptor inhibits RAG, and again it degrades all the remaining possible looped out DNA and free proteins that are generated through the different recombination events. And this is really important because it creates a homozygosity in the B cell receptor for that particular B cell. So that B cell will be very specific to the antigen, both shape, size, and sequence of that particular epitopic effacing amino acid strain, the continuum on a certain protein. But again, this is where B cell receptor and T cell receptor are a little bit different. B cell receptor is typically gauged towards effacing amino acids, but actually can develop to have certain amino acids which are also capable of recognizing certain residue decorations on amino acids. So things like glycosylation or lipolation. If you have a lipid, a lipoprotein, or if you have a glycoprotein, things like that, which then can be downstream conceived that some antibodies can actually recognize lipids and sugars, but to a smaller avidity than they do to actual proteins. Simply because amino acids on the antibodies are going to bind to amino acids on proteins. But given certain chemical makeup, they can actually recognize monomeric lipids and sugars as well. So small pre-B cells, again, they produce a fully functioning, oh no, sorry, a fully stable, I don't mean to use a functioning. So a fully stable heavy chain, and again, if it's enough to make the V pre-B and lambda 5 pre-B cell receptor, it's going to turn off the VDJ recombination in the successive chromosomes of the heavy chain loci, and this will also in turn activate RAG and direct it to the light chain again, because there's going to be no more sequence in the heavy chain, and there's going to be a series of signals that then shift RAG and its ability to bind DNA to the light chain. So as this moves along, when we get combination in the light chain, again, this is just V and J, RAG is reactivated. So, it's active. We make the heavy chain, it's turned off while it's being tested for stability. Once it's stable, RAG is turned back on. And again, because of changes, both epigenetic and insulator sequences that have now been bind to transcription factors, the heavy chain is no longer able to bind RAG. This then moves RAG and allows for the V and J recombination to occur in the light chain. If for some reason the first segment produces a non-productive V and J, it can actually keep on, which is different than what we saw with T cells. But in the light chain of the B cells, you can get multiple successive rearrangements of the V and J segments in relationship to the development of the light chain. Now, this is kind of unique because, again, this not only occurs in one chromosome and then to the second chromosome, but it also occurs through two different distinct loci. It actually tries through the kappa loci first, and if all of the rearrangement possibilities that randomly assigned in the kappa segment don't function on both chromosomes, then it also occurs in the lambda locus. And again, it will go through the first chromosome of successive recombinations, and then we'll go through the second chromosome successive recombinations. So this is why, even though the majority of T cells fail this system because of the way the T cell receptor is developed, the B cells need a even broader breadth of identity because of their ability to not only have antibodies that recognize proteins, but also antibodies that recognize other macromolecules like lipids and sugars. That you need different types of recombination events in order to develop, evolutionarily speaking, the right combination of amino acids on the variable segments on the recognition loops in the variable region in order to recognize the different amino acid cassettes that you're going to come across with proteins, but also the different other macromolecules that antibodies can bind. So this is why you have basically a successive series of different recombination events that occur in two different loci on two different chromosomes. So you actually get a multitude of recombination events that happen over multiple chromosomes that then can also happen over the two different loci. So this gives you a very distinct advantage of widening the breadth of recognition of B cells and their B cell receptors. So as we move from the B cell, the pre-B cell into the immature B cell, right, we have produced a stable, we have a stable, a heavy chain. We have a pre-B, a pre-B cell, the V pre-B and the lambda 5 surrogate alpha chain, or light chain, excuse me. And so basically, we get successive rearrangements in kappa that occur, and if it's productive, it results in the production of a kappa-based rearrangement and in respect to a heavy chain and a light chain that is kappa. If it doesn't, it moves to the second chromosome. And again, this will go through successive, multiple, not just one or two, it will go through 5, 10, 15 different rearrangements on the second kappa chromosome. And if that is successful, you still end up with a kappa light chain to a already produced heavy chain. If both kappa chromosome loci fail, you get successive expression from the lambda, and lambda then expresses from the first chromosome. Again, successive recombination events. If it fails, you get successive recombination events in the second lambda. And if all of these different things fail, and you have to think about how many different places this has an opportunity to work. But again, different recombinations are going to loop out different V and J and segments over time. So if you're looping out the ends, that's only going to produce or only have the ability to produce one possible recombination event. But if anything happens in between, it's however many variable segments and joining segments you have remaining, after everything else is looped out in between pulling those genes together. So if you have blocks like so, and you're looping in, you know, blocks like so, if you're looping this segment to this segment, only this is then going to be looped out to produce that to produce that successive recombination event. However, after uh, after this occurs, you can get second activation of RAG, which will then loop out the remaining segments in that region. But until that B cell has produced a functioning B cell receptor, you don't get any other looping out other than the loop outs that are required in order to join a V segment with a J segment. So anything in between that those V and J segments gets looped out. Anything on the periphery can still be utilized as successive recombination events in order to try to develop a functioning B cell receptor. So here it is, kind of in kind of grand kind of view. The early pro B cell begins to produce V pre-B and lambda 5 as it's rearranging the D and J. So 1 is D and J, and then 2 is V to the D and J segment of the heavy chain if it rearranges and produces a stable heavy chain. Heavy chain is going to then bind V pre-B in lambda 5 in the first checkpoint, which is going to then produce or send signals to then produce Ig alpha and Ig beta and turn off it turns off VDJ recombination and turns on VJ recombination of the light chain. Then you have the second checkpoint, which is again, the removal of V pre-B and lambda 5 in favor of the VJ recombined light chain to make a functioning B cell receptor. If the B cell receptor is productive and can signal, so it's functional and stable at this point. So you're looking for functionality and stability at this point. It then becomes an immature B cell. If any way along the way of the first two checkpoints, if the heavy chain does not create a functioning, a stable heavy chain protein complex, the solubilized V pre-B and lambda 5 as a pre-B cell B cell. So as this particular B cell developmental stage that does not have a B cell receptor, the cell dies by apoptosis. And again, if you make B cell receptor heavy chain that's stable, but after all of the different recombination events in the light chain, it fails, that cell then also apoptosis. So even in the beginning, just like we saw with T cell receptor, we look for stability and then we look for function based on the ability to combine both a heavy segment and a light segment. For B cells, this was the difference between the beta and the alpha chain, right? Beta was the heavy, alpha serves as the light, or the gamma delta if that outcompetes the beta rearrangement. And for B cells, we don't have that same kind of heterozygosity in our chains because it's just one heavy and one light chain with no two distinct other loci for each individual chain, just the chains themselves. So this is what allows it to happen. So when the pre-B cell receptor undergoes the transition where it binds the light and heavy chain, this then binds the kind of co-functioning, I won't call it a co-stimulating, but I'll say co-functioning receptor alpha and beta, sorry, immunoglobulin alpha and beta subunits that combine to they bind to both the constant region and have co-localized cytosolic components that create a receptor known as CD79 And so CD79 plus B cell receptor in its entirety is what makes a immature but naive functioning B cell receptor. So as I told you before, there's a number of different genes that kind of help transition the B cell from stem cell all the way to a mature B cell. And one of the most important of these is PAX5, which is all the way down here. So PAX5 and E2A and EBF, again, which regulate PAX5. They also regulate a number of other genes. They basically are non-existent in, if we follow this along from this standpoint, right? They're not present on stem cells, but when we move to early pro B cells, and this is where we're starting to get IL-7. So this goes back to IL-7 and CD127 expression. This is what helps activate E2A and EBF and PAX5, and their expression is actually on continuously all the way through, and not even a change in expression density from late pro B cell all the way to mature mature B cell. Now there are different markers that are expressed in different stages. Really importantly, something like KIT, which is expressed to help again define the early pro B cell as it moves on from being a stem cell. It comes on at the same time as IL-7 receptor, which again, IL-7 and IL-7 receptor expressed at the same time as E2A and EBF, as well as PAX5. And all of these really play an important role in kind of space defining um, in more temporal defining function, because these are all important for, again, the early generation of the pre-B cell receptor. So the activation of the pre-B cell receptor starts by expression of IL-7 and KIT, as well as E2A and EBF, PAX5, because these genes help regulate RAG and TDT, which was the terminal deoxynucleotide transferase, which helps us enclose the VDJ segments in a non-additive function, like we saw with T cells. So this is what's going to generate our normalized somatic mutations in the joining segments of DNA between the bigger segments of DNA. And again, this is where, in the function of TDT as well as RAG, is really where the ability to produce a stable versus functional B cell receptor is really important, because stable just means producing an in-frame B cell receptor. So like, do you have a B cell receptor that's the right size, that has productive amino acids in a way that produces the shape and in homozygosity that a B cell receptor is supposed to have, as well as a really important question, which is when you have these random mutations that happen through TDT and non-homologous end joining because of the function of RAG, the first question is, do you actually make a B cell receptor, the heavy chain, that actually looks like a heavy chain, so just the architecture sequence and stability makes sense. That's a different question than function. And so stability is the first thing, just like we saw with the T cell receptor. The function then comes in, because again, proteins functions based on structure. So the first thing you gotta figure out is if the structure is right. If the structure is right, that also doesn't mean that the function is correct. So first start with structure. If the structure is great, then you move on to function. And this is all because this is all random. How the sequences can be combined is random, and how TDT and non-homologous end joining somatically mutate the joining segments between the VJ and VDJ segments of recombination. Again, if they're not a factor of 3, therefore shifting the frame of that sequence, you're not going to produce a functioning thing. That's checkpoint one, and checkpoint two in the grand schemata of B cell development, um, is whether or not the B cell receptor actually works, and that's going to become a kind of a thing of, um does the sequence that you've created, um be able to recognize the antigen and not, and then part two, which is, does it not recognize human antigen? So there's a lot to unpack here, but I'm hoping that this kind of all makes sense. Um, again, and I kind of wrote this down here because I think this makes it a little bit easier. So PAX5 again regulates the expression, because it's related to E2A and EBF. E2A and EBF is based in the expression of the VDJ recombination of the heavy chain. So heavy chain is created, it's stable, this activates E2A and EBF. This then activates PAX5. They come together to activate lambda 5 V-pre-B to make the pre-B cell receptor, and CD19, which is going to serve as the B cell co-stimulatory molecule. This is the alpha and beta chain. And then beyond all of this, so again, these all kind of stimulate at the same time. So here in the early pro B cell, so early pro B cell, you know, you're getting the beginning of activation of KIT and IL-7, but then this is when you get in first expression of RAG, first expression of DDT, first expression of the V-pre-B and lambda 5, CD19, and then yeah, sorry. So CD19, immunoglobulin alpha and beta, all of these normal things are all starting at the same time, and they all are expressed throughout the life of the B cell, except for CD43, which is a differentiation marker, and then and then BP1, which will become important later as a differentiating B cell specific gene. And then we have one gene that we haven't talked about, and this is prudence kinase, so BTK. So BTK is going to serve as a really important kind of co-molecule. So again, if we go back real quick, too far. Where's so again, we have the B cell receptor. B cell receptor, again, has a heavy chain, has a light chain, and it has this kind of co-receptor, not co-stimulatory molecule, but this co-receptor, immunoglobulin alpha and beta. And then there will also be a co-stimulatory molecule later on, which is CD19. Um, yeah, which is CD19, and CD19, CD45, and PTK are all really important in transitioning the B cell from the early pro B cell to the late pro B cell. And that transition is really important because that is what allows for the activation of RAG in order to undergo VDJ recombination. And then the expression of these genes also regulates the expression of RAG in a way that it gets shut off while the large pre B cell is interrogating the structure and stability of um, the heavy chain. And then when the heavy chain is stabilized and makes uh, the pre-B cell receptor in combination with CD19 and immunoglobulin alpha and beta, this then triggers the transition to a small pre-B cell. The small pre-B cell then undergoes VJ recombination of the alpha turn out of the alpha uh sorry of the keep saying alpha, what I mean but say is the light chain. The light chain then produces either does or does not produce a functioning IgM copy or version of that particular B cell receptor. And if it does, this is what turns RAG off, and you get functional production of B cells. And again, a lot of these are the genes that help push the B cell on its trajectory are all kind of shut off around the time where VJ recombination occurs um, in the light chain, including expression of the surrogate light chain lambda 5 in V pre B. But other genes, like a lot of the signal transduction genes and the transcription factors, are continuously expressed all the way through. And BTK, as we'll see later, is really important in not only development of the B cell, it's also going to become important for B cell activation. And in the absence of BTK, that's actually a really well studied and really well known immunological disease, is called Bruton's kinase X-linked 8 gamma globulinemia, because without it, B cells cannot develop into mature naive B cells that can then create antibodies. So here is BTK in what it does. So BTK plays an important role in signaling through the B cell receptor and CD19. So here's CD19 as its as a co-receptor here. Here's B cell receptor. Here's immunoglobulin alpha and beta. And here's BTK. So BTK is activated through B cell receptor CD19 co-stimulation that activates the Lin-Sic pathway. And Lin-Sic phosphorylation activates BTK. BTK will eventually activate or phosphorylate Blink, and Blink then actually activates a great number of other genes, including regulation of RAS, RAF, ERK. So this is the MAP kinase pathway, as well as the DAG IP3 network, again, which activates MAP kinase, AKT, which all play an important role in expression of things like NFAT, transcription factors related to B cell development, as well as NF-kappa B, because what we've seen before. So here's going to have another look at it. So when you have co-stimulation CD19 in immunoglobulin B cell receptor, this activates BTK. BTK activated both through the Lin-Sic pathway itself. BTK phosphorylates and activates phospholipase C, the DAG IP3 network, activation of MAP kinase and junk. So again, proliferation, protein synthesis, AKT activation, and then BTK phosphorylates the inhibitor of kappa kinase. This is again phosphorylates inhibitor of NF-kappa B. NF-kappa B, the inhibitor of kappa B is lost, right? It's degraded, and this releases NF-kappa B. This allows for the activation of B cell specific genes again. So we're going to get to this place where B cell specific genes is really important for, again, to go through the development and activation of the different segments of the immunoglobulin locus, but also for the expression of B cell specific cytokine and chemokine expression as the B cell develops, and then again, once it's in circulation, when it's going to activate. So these same genes have a lot of crossover in order for pushing the B cell through development, as well as the function of B cells when they're called upon to function. So, interestingly, right, all B cells, so all cells except for B lymphocytes, the immunoglobulin locus is closed, and this Ig locus is very different from the immunoglobulin locus that's found in the T cell receptor zone. So the T cell receptor immunoglobulin locus is on a different chromosome than is the B cell immunoglobulin locus, because there's a different series of VDJ possibilities. Now, the only thing that is the same is that the RAG genes and the RAG recognition sequences in between each different segment remains the same. So you don't need to have RAG specific for T cells and RAG specific for B cells. It's all the same RAG and all the same function. So when B lymphocytes are developing, the immunoglobulin locus in the chromatin is actually phased in the closed position. But then when PAX5 is activated, again, this is why it's really important as a transcription factor. This binds to an enhancer way in the 3' end. So this is the 3' enhancer. So this is way downstream of the actual promoter. And you know, canonically speaking, you think of binding to upstream elements in cis when you're going to phase the chromosome, the chromatin in on position. But in this case, the enhancer actually exists way upstream. And this actually forces open the FC region. And when the FC region opens, this causes a cascade or a domino effect that then allows for the sequences in between the D and J and the V segments to open, so they can be recognized by RAG. So you get, again, a binding of the enhancer by transcription factors in the 3' end. This opens up the sequence to allow for DJ to recombine. And when the DJ recombine, this then allows, again, a cascade of kind of, it's polar, right? So it's pulling the chromatin along as it's recombining. This then opens up the V segment that's already been bound to transcription factor. This then, again, opens up the DNA in the recognition sequences for RAG. This causes looping, creating the VDJ kind of loop. The VDJ loop is then again looped by way of binding an upstream enhancer to the mu segment, the mu C terminus or FC region that still has the signal sequence. And the signal sequence for mu, again, is the sequence that's going to put it's what puts the B cell receptor in the membrane. So the signal sequence, so here's the signal sequence. It's on this kind of component here. And so it's what actually designates the B cell receptor to be specifically put in the membrane. When the signal sequence is looped out in favor of solubilizing the antibody, when this becomes a plasma cell, this will undergo one more recombination event where the signal sequence is actually looped out, therefore only creating a loop with one FC terminus. And then that's what, without the signal sequence, there's nothing to hold the antibody in the cell. And so it's released from the cell and solubilized. So this is kind of an important function of B cells, because B cells, because they are created throughout our entire life, there's regular breakage and recombination events occurring, especially in the immunoglobulin locus. And this makes B cells really a really key cell type to a large number of different kinds of tumors. One of the most common of these is something called Burkitt's lymphoma. And Burkitt's lymphoma is typically associated with a viral infection called Epstein-Barr virus, or EBV. And the thought is that EBV is very specific to salivary cells and to B cells. And once it gets into B cells, because it's constantly inside of the B cells, it might continuously ask for recombination events to try to get at the virus, because there's always that underlying signal that the virus has invaded the cell. And this results in a few cases, not in the great, there's over 98% of the world's population is naturally infected with Epstein-Barr virus. So this happens in a very rare subpopulation of people infected with EBV. But in the right set of circumstances, chromosome 8 and chromosome 14 translocate to one another, which actually puts the MYC gene, which is the gene, one of the major genes that's involved in embryonic cell proliferation. So it's a really powerful proto-oncogene under the regulation of the Ig locus. So the transition from the MYC gene onto the so from 8 to 14 that then puts MYC under the control of the immunoglobulin gene locus is then going to just mass produce MYC because the B cells are always undergoing these recombination events. If you have a B cell, they're always undergoing these recombination events because, again, you're always making B cells. This is not like T cells that have a limited capacitance for development because of the age or involution of the thymus. So if you have Epstein-Barr virus and Epstein-Barr virus is inside your B cells and you're getting constant breakage and rejoining of the immunoglobulin locus, all it requires is a secondary nick of the DNA of one of these genes or another proto-oncogene like BCL2 or other anti-apoptosis gene or cyclins that would result in the form of lymphoma. So so other types of B cell diseases can arise from B cells. So we have again, there are different kind of B cells basically as you develop. So as a fetus, and when you're in mother's womb, you develop B1 cells. These B1 cells have a really poorly restricted ability to undergo VDJ recombination. And so, there's not a lot of high production of great isotypic switching, which again is why things like vaccines for late stage pregnancies for the production of IgG and the production of IgA through breast milk secretions is really important because after birth, again, this takes time, but you do eventually get conventional B2 cells, and these are the type of B cells that actually are the B cells that you specifically think of as B cells because they have very directed isotype switching based on the type of infection. And this is because they require T cell activation and can undergo hypersomatic mutation in order to develop memory. So in totality, this kind of blows up all what we've just been talking about. Again, between early and late pro B cells, like the pro B cell is really just about rearrangement of the VDJ heavy chain production of a stable, right? This is what we're looking for, stability, a stable heavy chain, and it's a stable heavy chain that undergoes not only rearrangement but also it undergoes TDT somatic mutation, not hypersomatic mutation, but somatic mutation. And this is why, because this all happens at random, um, and it only happens based on single rearrangement. Yeah, right. So you can have random success. Sorry, not single, but you have successive rearrangement. So successive rearrangement of the heavy chain over the two different chromosomes. If it fails, the cell apoptosis. And then again, if this works, we get again the activation of V pre B and lambda 5, as well as CD19 and immunoglobulin alpha and beta. So this is co-stimulatory molecule, this is co-receptor, and then this is surrogate light chain, right? And the signals that come from this, plus again, the activation of PAX5 and the other transcription factors then allow for the reactivation of RAG, which again, in successive order, will go through kappa. If kappa works, you get mu and kappa expression. If you have lambda that works, then you have lambda and mu expression. If all successive rearrangements fail, the B cell undergoes apoptosis. So once the B cells are created and produce a productive and stable B cell receptor that are, again, IgM to start with, B cells are then tested against the bone marrow expression of self antigens. These are typically just regular bone peripheral proteins. So these are membrane bound proteins, glycoproteins, lipids, glycolipids, lipoproteins, etc. And you're looking for again the inability to recognize self antigen. So when there's no expression of self antigen or no reaction to self antigen, the immature B cell is then moved into the blood and expressing IgD or IgM, often than not IgM, that is still membrane bound. If there's a reaction to self antigen, something really interesting happens, and that is that the B cell is then retained in the bone marrow. Now, the reason that this is because B cells, unlike T cells, can undergo one other remaining segment VJ rearrangement as long as it has not been looped out. So if this is either in the middle of the successive rearrangement in the first chromosome, or in the middle of at the end of it, and it has occurred on the first chromosome, but it hasn't undergone the light chain rearrangement on the second chromosome, on either kappa or on lambda, depending on where it is. If it's anywhere along this line that has not completely moved out all the V and J segments in the DNA, the receptor can actually, the B cell can be resequestered in order to see if a production of another recombination event occurs. And again, this is through you know this is through a series of different signaling sequences. So when you have binding to self antigen in this microenvironment, this then triggers a different cascade of phosphokinase relay system that then reactivates PAX5. Sorry, I'm running low on this. E2A and EBF that reactivates the VDJ or the VJ recombination events in whatever successive segment you're in order to reproduce the light chain. Because again, the recognition of a B cell receptor is a combination of the variable segment of the heavy chain in combination with the variable segment of the light chain, in how the CDR3 joining loop plus CDR2 and 1, as you come out along the horizontal plane, can recognize the antigens that the B cell is being interrogated for. So if there's still a chance for receptor editing to occur, it does. If, however, it cannot be rescued, the B cell is either then committed to apoptosis or effectively becomes senescent due to a non-peripheral tolerance mechanism. Now, the interesting thing how this works is, again, if you have the bone marrow cell and it recognizes antigen, the immature B cell continues to keep rearranging, very specifically the V and J segments. But again, when it takes out a segment to then religate new segments, all the internal segments in between the two are now gone. So you get this random resequestering, and the thing is, you can't rejoin ones that have already happened. So for example, this J segment and this variable segment is already used up. It cannot be reproduced. And there are transcription factors that bind in cis that prevent this from recombining. And so then it's a matter of random, allocation of RAG binding to a J segment and to a V segment. And as they loop in, the rest of it's looped out. And again, if that occurs, you make a different B cell, and because of the light chain, because it's all light chain differentiated, it's going to have a different specificity. But that different specificity may be close to what the old receptor had, or it might not be. So as the immature B cell makes the new light chain, if the new light chain is now self-reactive, again, you can continuously keep, disengaging from those antigens and try to undergo rearrangements. But again, once you have completely used up all your V and J segments and there's nothing else remaining on either chromosome or in the successive line of VJ, in both lambda and kappa, or kappa and lambda respectively, then the cell probably apoptoses. Now, if the B cell is released from the bone marrow and still can recognize antigens on cells, because again, they're just recognizing the repertoire of antigens on bone marrow cells, which is extensive, but not everything, right? This is not AIRE or AIDE assisted antigen recognition. So there are B cells that are going to be able to recognize self antigen or soluble antigen. This is where B cells are signaled to become unresponsive and become anergic. And so as they bind to self antigen, again, this really happens because of the lack of danger signals, lack of previous injury, or this lack of damage, and the lack of damage results in a successive, basically oversensitization of receptor that can recognize either soluble or cell-bound antigen during initial release. And if that happens in the absence of damage, the B cell undergoes a non-contact dependent energy, a non-contact dependent energy, which is different from what we saw with T cells, which is contact dependent, and that's mostly because, again, T cells cannot recognize soluble antigen, whereas because they had to find it bound to MHC, whereas B cells are not looking for MHC, they're looking specifically for soluble or membrane bound antigens. So once the B cells come out of the marrow, they exist as immature B cells, and they must become mature before they can actually be possibly activated in the case of an infection or an immunological cascade. So in this case, as B cells develop, there's a underlying or an undercurrent of expression of CCL21 and CCL19. Again, this is lymphocytic specific, because CCL21 and CCL19, these are going to increase homing receptors specific for lymphocytes to get into the lymph node. Now, the interesting thing is these cytokines are received by CCR7, so chemokine receptor 7, which is a G protein coupled receptor. And it, just like T cells, it recruits B cells to the secondary lymph, and it's actually the dendritic cells inside the follicles of the lymph node, which continuously express CCL19, and CCL19 then allows B cells to enter through the high endothelial venule through the bloodstream, and then they then are recruited up to the germinal centers, or again, this is the follicles uh of the lymph node, which are known as the B cell area. So it leaves the T cell area and then they reside specifically in the B cell area. And they can transition between the two in this route, leading towards the efferent vessel or remaining inside of the lymph node, takes the B cell through the T cell zone. And as we discussed in class today, that's because T follicular helper cells, which exist in this borderland, are really important for the activation of B cells. So of course they have to pass the T cell zone in order to not only mature, but also to then be able to activate. So so as they enter into through the high endothelial venule, the follicular dendritic cells secrete one more cytokine, so they express CCL19 and CCL21. But then they also excrete CXCL13, and this is then received by a receptor on. So here is CXCL13, and only B cell type cells express the CXCL13 receptor. And so this is what allows T cells to be honed to the lymph node, but stay inside the T cell zone. And the B cells then move their way specifically to the follicles. And then the B cells then express lymphotoxin LT, which is similar to TNF alpha, and the cells in the follicles produce BAFF, which is B cell activating factor, and these two actually uh, allow for not only the recruitment of B cells to the follicle, but then this moves the B cell into becoming a mature but naive B cell, in which then can then circulate through the bloodstream into the lymph system, again waiting for the call of a hyperexpression of CCL19 and CCL21, which will draw them back to the lymph nodes where they will wait to see if they'll be activated. So when B cells are actually activated or when they're selected for, so again, if you have an infection, just like we saw with B cells, when you have the infection, CCL19 and CCL21 will draw them to the lymph node. CXCL13 will draw them back to the germinal center. And if they are a B cell that is selected for to be activated, they'll pass back to the T cell zone where they'll interact with T follicular helper cells and T helper cells together. And the cytokines secreted by T helper cells and the activation by T follicular helper cells then give you an antigen or epitope specific differentiation into centroblasts, and then centroblasts into plasma cells, which lose that C terminal signaling sequence, which solubilizes the B cell receptor into antibodies. And then these reside in the medulla of the into the medullary cavity uh, of the lymph node, and then either move out or secrete antibodies into the efferent vessel, which then get deposited into the bloodstream. All told, the continuing division of plasma cells and during the course of an infection actually results in being responsible for about 20% of all the secreted proteins inside of the blood during an infection. And the B cells that remain inside the germinal center that don't become plasma cells are the ones that will take on the moniker of B cell memory cells. So we're gonna talk about B cell tumors, because hopefully we'll get to B cell tumors when we talk about immunodeficiencies and dysfunctions. And that does it for the B cell development. I hope you guys enjoy, and let me know if you have any questions.